All right, so in the previous class, we had discussed up until the viability of the pollen. Now, did you people have any doubts? Anything that you wanted to ask me?
No, ma'am.
Sure?
Yeah.
Okay. Okay, so now we're going to start with one more thing, that is cryopreservation. Now, what is cryopreservation? See, just like, have you ever heard about seed banks? Wherein a very unique seed or unique plant seeds are stored so as to, they can be used to grow a new plant later on. So that is a seed bank. Now, same way, the pollen grains can also be stored. Okay, preservation of pollen grains. So, cryopreservation, it is the method of storage of pollen grains for years in liquid nitrogen. This is a chemical that is used at minus 196 degrees Celsius. At such cold temperature, the pollen grains are stored, and such stored pollen can be used as pollen banks. Now, for example, the pollen of certain plants have to be transported somewhere else. But in that transportation time, the pollen will not be viable anymore. So those pollens are stored in liquid nitrogen. And when that happens, what happens is that the pollen grain stops growing. Okay, it gets arrested for that period of time. And when it is taken out from that solution, then it will be viable. And this type of technique wherein the pollen preservation has been done, it is used in crop breeding programs. What happens in crop breeding? What we do is, like, if there is any desired characteristics that we want in a crop, okay, so we'll use that particular pollen. Like, normally the plant will not actually reproduce that way, but when we add certain pollen such as that, then a different cross is made. For example, we want to, it's not possible, but just for the sake of example, if we want to cross a hibiscus with a rose to see what is the new flower that we get. So then, if we want to keep rose as the male, then we have to take the pollen from the rose. If we have to keep the hibiscus as the male, I'm just saying this as an example. This is not a valid thing, but it is just to give you the idea as to when these will be of use to us. Mostly it is used in the crop breeding programs. Clear? And also for the times, like for example, in one place there is the certain climatic condition in which the plants grow, okay, by the pollination and everything happens. But then in another place there is no condition. Now the pollens if of this place will be stored, then it can be transported here where it cannot be available naturally. So that is also very helpful because of this method. You must remember two things. The first thing that you must remember is liquid nitrogen. Second thing you must remember is minus 196 degrees Celsius. This is the temperature at which the storage of the pollen grains is done. This much is clear?
Yes, ma'am.
Okay. Now we will discuss the gynosia. Okay, there is some part that you people still have to note down, right? Okay, I'll come to that. Let me explain one topic, then I'll give you time. Okay, now we have discussed androecium. What is androecium? The male reproductive part. And what is the unit of androecium? One unit of androecium.
Anther and filament.
Anther and filament together are stamen.
Oh.
Oh, so stamen is stamen is made up of anther and filament. Now we'll talk about the gynoecium, that is the female reproductive part, and the unit of the gynoecium is pistil or carpel. Now, in any flower, when there is only one pistil present, okay, one of the pistil is present, then that type of flower will be monocarpellary. When only one carpel is present, okay. Then if two are present, then bicarpellary. But more than two or more than one, then we'll call it multicarpellary, depending upon if the flower has one carpel, two carpels, or multiple carpels. Okay. Now, when the pistils, whenever they exist in two kind of conditions. One is in a fused state and one is in a free state. Now, if you look at here in this particular diagram, the first one, you would see that all of them are joining at one point here. You seeing this? This type of pistil is known as what? Syncarpus. Okay. Syncarpus means, syn means fusion, right? Syngamies. Right? Syn means fusion and carpus is referring to the carpal condition. So syncarpus. This is found in papaver. There's a plant which is known as papaver, and this is very important. Syncarpus type of pistil or ovary is found in papaver. Now, if you look at the second part, you will see that all of it has free ends. None of it is joining another. All of the ends are free. So this type is known as apocarpus, wherein the carpals are free. And this is found in mycelia. So papaver and mycelia. Two examples to remember, very important. Okay, this is clear. Now this is the differentiation of the pistil or the carpal depending upon whether they are fused or whether they are free. Earlier, the differentiation was because of the number. When the carpal is 1, that is monocarpillary. When the carpal is 2, then that is bicarpillary. And when the carpal is multiple, then that is multicarpillary. Clear? Now this thing, so you already know that the cells in the body are of two types. One is the somatic cells. The other is the germ cells. The somatic cells are the body cells, or we can call them as the vegetative cells. And the germ cells are the reproductive cells. Always remember this thing, okay? There is a difference, and the somatic cells are the majority cells of the body and the germ cells are less in. Clear? Now we'll talk about, just like how stamen had two parts, anther and filament, the pistil has three parts: stigma, style, ovary. Stigma, style, ovary. Now, stigma is the landing portion. See, let me just clean this. You see here, this part, this is the stigma, sort of like a platform. It is the landing portion, stigma. Then elongated, landing portion for what? So for pollen grains. The pollen grains that will be dehisced, that will be released, they will come and land here. If this is a pollen grain, it will come and land over here. Okay. Now, this long portion that we are seeing, this is the style. Okay, that starts from the stigma. Elongated means long, slender means thin. So it is an elongated, slender part, starts from beneath the stigma. Lastly, you're seeing here a very swollen portion, right? It is swollen. So the basal bulged or swollen portion is the ovary, and it is present at the base because basal. Clear? Now, inside the ovary, ovules would be present, but not so easily. There is a whole entire thing that we have to go through. Inside the ovary, ovarian cavity is present. This ovarian cavity can also be called as locule. If you say that inside the ovary, locule is present, then also it's fine. If you say that inside the ovary, ovarian cavity is present, then also it's fine. It's the same thing. So inside the ovary, ovarian cavity or locule is present. Now inside the ovarian cavity, placenta is present. The placenta of the plants is present inside the ovary and inside the ovarian cavity. Now, the megasporangia, such as there was microsporangia, right? The megasporangia or the ovule, they would arise from this placenta. So all in all, we can say that ovules are present inside the ovary, but not so easy, isn't it? There is a stepwise way we have to reach the ovule. So now, this is how, like, inside the ovary, there is ovarian cavity. Inside the ovarian cavity, there is placenta, and this placenta gives rise to the megasporangia, that is the ovule. Now, ovule is not the fancy name, megasporangia is the fancy. Ovule is just normal terms, but if you want to, refer it professionally and formally, then that is megasporangia. Okay. Now, the megasporangia forms the megaspore, that is the egg. The female gamete, just like how the male gamete was microspore, pollen, right? Same way, the female gamete is megaspore, that is the egg. Clear?
Yes, ma'am.
Okay. Now see. Ovule develops into the seed. The seed, like for example, mango. Okay, everyone has seen a mango, eaten a mango. The seed of the mango is the ovule. Okay, and the whole entire flesh along with the peel, the flesh, the part of the mango that we eat, that part, and the peel that we do not eat, that is the ovary. So the ovules are present inside the ovary, just like how the seeds are present inside the fruit. The fruit is the flower's ovary. The matured ovary of the flower is the fruit. The matured ovule of the flower is the seed. Okay. Now, we know that mango has one ovule, means mango has one seed. And can you tell me the example wherein multiple ovules are present?
Watermelon.
Watermelon, correct. Watermelon, papaya, right? So many seeds are there. So those seeds are ovules. Always remember this one thing. Ovary is equal to fruit. Ovule is equal to seed. The thing that we throw out is the ovule. We don't eat the seed, right? So that is the ovule. Now, in many fruits, there will be just one ovary, such as in wheat. Wheat has one seed. Paddy, that rice has one seed. Mango has one seed. Okay, so this is one ovule. And remember one thing, in the world of plant, everything is a fruit. The thing that we consider as vegetable is also a fruit. Why? Because it is a product of sexual reproduction, right? So don't confuse like why ma'am is calling wheat as a fruit. It's definitely not a fruit in terms of English, but in terms of biology, in terms of botany, it is a fruit. Because why? Because it is a product of sexual reproduction. Always remember this one thing. Remember it as a rule that in like, as far as the angiosperms, meaning the flowering plants are considered, the product of sexual reproduction will be seed and fruit. Mostly seed and fruit. Clear? And then when the number of ovules in the ovary are multiple, then we have papaya, watermelon, and even orchids. Okay, again, orchid is a flower, right? So it varies. Now, this much is clear to you people?
Yes.
No. Okay, very good. Now, moving forward, there is megasporangia. But first you note down, because you know the third student, Yahya, is not here. He'll be joining at like in 5 minutes. So up until then, you people note down, okay? From where do you have to note down? I think from here, right?
Yeah.
Species. See, I drew such hideous things to remember it. Write slowly, slowly. Don't write so fast. Okay, till then you people can switch off your video, write comfortably, and then when I'll teach, then you switch on.
Done.
Both of you all done? Okay.
Done.
Okay. Hello, Yaya. How are you? See, I am right now. I've given you people time to note down. So you also note down the thing that you had to from the previous class, right? From here. So just note down and tell me when you're done. Okay.
Okay, ma'am.
Done, ma'am. Can you scroll down?
Done? Shall I scroll?
Yes, ma'am.
Done.
Done, ma'am. Ma'am, can you go up for a minute?
Done, ma'am. Done.
Also, you should draw this also. This is important. You should know where to label, how to draw it, because mostly it is asked in questions also, like draw the cartwheel. So you should draw this, okay?
Done, ma'am. Done.
All right. Okay. So I had discussed up until here, right? Okay, you complete this part is left. Everyone else is done?
Yes, ma'am.
1 minute, ma'am.
Sure, I'm waiting.
Done, ma'am.
Okay, so now we'll discuss this thing, the megasporangium, that is the ovule, okay, or the megasporangia. Now see, the ovule is of prime importance as far as the reproduction in plants goes. Okay, so now the ovule, it is a small structure. Now we know that it develops from a placenta. So this is a small structure which is attached to the placenta with the help of a stalk that is known as funicle. See this thing. Now we'll look at the diagram and see each of those labeling and understand that. Okay, now rest of the students, switch on your videos. Now see this. The part which is actually attached onto the placenta area inside the ovary is the funicle. This will allow the ovule to get attached. Okay, so this is sort of like, imagine it like an extension which is there to attach it. Now, The region where the body of the ovule fuses with the funicle is known as the hilum. Now, this part that we are seeing, it is not the part of ovule, it is the stalk funicle. Now, the part of ovule starts here, and the part wherein the ovule is fusing with the funicle, that is the stalk, is known as the hilum. So, hilum is the junction wherein the ovule fuses with the funicle. Okay. So we can call hilum as the junction between the ovule and the funicle. It is attaching it together. Now, the ovule has protective layers surrounding it, and those layers are known as integuments. Integument means, in general, it means layer. Okay, so the layer surrounding the ovule is the integument. Now, this, see, integument is present everywhere. Like, okay, since it is the protective layer, so it is present everywhere surrounding it. But what happens is that there is one portion wherein the integument is absent, and that portion is the micropyle. So remember this one thing, that micropyle is the small opening wherein the integument is not present, because it is a pore, it is an opening. So if the opening will be covered, then there will be no opening, right? So that is why micropyle is the area wherein the integument is absent. Now, micropyle is the small opening from where the pollen tube enters the ovule. We'll talk about pollen tube later on, but just for now, you should understand that micropyle is the pathway through which something will enter. And at that point of area, there is no integument present at all. This much is clear. The different parts of the ovule are clear, such as, first of all, the funicle, secondly, the hilum, then the integuments, and lastly, the micropyle. This much we have discussed up until now. Now, see, the micropyle end, okay, it is, even though it looks like that, and we'll imagine it, this part being the downward part, right? But in terms of the plant, this is just for us to understand. But in terms of the plant, this part is the top part. Okay. And the opposite part is the basal part. Basal means the bottom part. So now, opposite to the micropyle end is the chalazal end. The chalazal end represents the basal part of the ovule. So right opposite to the micropyle, here is the micropyle. Right opposite to the micropyle is the chalaza. And the micropylar end is known as the micropylar pole or micropyle pole. And the opposite is the chalazal pole. And this is the basal part. Basal means the bottom part. Okay. Now, the mass of cells which are enclosed by the integuments, they are known as the nucellus. See this thing. This whole mass of cell is there, and this mass of cell is surrounded by the integument, inner integument, outer integument, and this mass of cell is known as nucellus, and it is very nutritive. It is the nutrient reserve. Okay, so you can call nucellus as the tissue that is high in nutrition. Clear?
Why is it called mass of cell?
Why is it?
Why is it called mass of cell?
Mass of cell because it is like multiple cells present there. So they're like, you know, like how a pile of coins, collective nouns. That is what it is, mass of cell. Okay. Now, the cells of the nucellus, they have abundant reserve food material. Since they are nutritive in nature, their sole responsibility is to provide nutrition, right? So they have abundant reserve food material. Just like how the mothers of a toddler, right? Whenever they go out for a park or picnic and everything, their whole purse is filled with snacks and everything. So the mother is the reserve food material, like she has it. And whenever the kid needs it, she gives it to the kid. So same thing, nucellus does it. Okay, nucellus has the abundant reserve. However much is needed, it will be provided by the nucellus. Clear? Now, inside the nucellus, embryo sac or female gametophyte is present. Male gametophyte, you remember, pollen grain? And then it divided and formed the two male gametes. Now we have embryo sac or the female gametophyte. And inside this female gametophyte, there will be the megaspore, that is the egg, which is the female gamete. Getting? The terminologies are different in plants as compared to animals, but you should understand that male gametophyte is pollen grain. Then it divides and forms two male gametes. Here, the female gametophyte is the embryo sac. See, this is the embryo sac. See, inside, the nucellus is surrounding the embryo sac, and this portion, this oval egg-like structure that we are seeing, it is not the egg, it is the embryo sac. And inside that, do you see this one cell? This one. Okay, this is the egg. This is the female gamete. Okay, all right. Now, so an ovule generally has a single embryo sac formed from a megaspore. Okay, so this is one embryo sac. Okay, and mostly the ovules have single this thing embryo sac. One embryo sac is present in one ovule. Okay, because if multiple will be present, then multiple male gametes will be needed, so as to not create that confusion and everything. There is just one embryo sac. Now this diagram is also asked many a times. It is very important to remember all the labelings that are there. Now see, read everything. Now you've understood all the parts of this ovule, and this is the diagram of an ovule. Okay, this whole entire structure is ovule. Funicle you've understood. Hilum. Then we have understood micropyle. We have understood integuments. See, integuments, two of them are there, outer and inner. So outer integument, inner integument. Then we have nucellus. Lastly, we have the embryo sac. And this chalazal pole is the basal area. Clear? Any doubts up until now?
Can you explain nucellus and embryo sac again?
Sure. See, nucellus is the nutritive tissue. Okay. So see, as I am saying that integuments are those layers which are surrounding the ovule. Okay. But more precisely, it is protecting the nucellus. Okay. Inside the ovule, nucellus is there. Now, nucellus is what? It is just, imagine it like cells like this. The cells filled with nutrients like this. Okay. Now, these cells are responsible for nutrition aspect. They have abundant food reserve. And they are surrounding the embryo sac. And why they are surrounding the embryo sac? Because when the embryo sac would develop, it would require nutrition. That nutrition is provided by nucellus. Clear? And embryo sac is the female gametophyte. Inside the embryo sac, egg will be present. Meaning, when the pollen has landed on the stigma, it will travel into the style, it will come into the ovary. Inside the ovule, it will come into the ovule. And in the ovule, it will come from here only, from micropyle, because this is the place, this is the hole. And then the male gamete will come and fuse with the egg. Okay, so this is why the whole entire thing is there. This much is clear? All right. Okay. Okay. So before megasporogenesis, you people noted down the structure of megaspore and say, yeah, and draw this diagram. It's important. The labelings you should understand. And since now all of you are in class 12, so just remember one thing: boards will be there, right? And in boards also, wherever in biology, wherever possible, you make diagrams. It's a rule. Okay. So you should practice the diagrams along with the labelings and everything.
Done, ma'am.
Done, ma'am. Done, ma'am.
Everyone's done?
No, ma'am. 1 minute.
Okay, okay.
Done, ma'am.
Done, ma'am.
Done, ma'am.
Now, moving forward, so up until now, we have discussed the structures that are associated with the female reproductive part and the male reproductive part. Now we'll move on to, just like how we studied microsporogenesis, now we'll study megasporogenesis. So obviously, the formation of a megaspore. Now, the process of formation of megaspores from the megaspore mother cell is known as megasporogenesis. Just like how there was pollen mother cell and microspore mother cell, same way here, the cell that will undergo meiosis is the megaspore mother cell, MMC. Now, the megaspore mother cell is differentiated by the ovule in the micropylar region. In the micropylar region, the ovule differentiates, means it divides and forms the megaspore mother cell. Now, this megaspore mother cell, it is a large cell which contains dense cytoplasm and a prominent nucleus. It has dense cytoplasm and a prominent nucleus. The megaspore mother cell undergoes meiosis to form 4 haploid megaspores. Just like how in pollen also 4 tetrad was formed, here also 4 haploid megaspores will be formed. Just like in that case also, the pollen mother cell was developed, then that underwent or undergo, what is the word? Undergo or underwent? Undergo, right? But if I'm saying it in past tense, what will it be? Underwent? I don't know.
It's undergoing.
Undergoing? No, undergoing is continuous. Like, see, at that point of time,
Underwent, ma'am.
underwent, is that a word? Because I have no clue. I feel like that's not a word. But anyway,
I think it's undergone.
it's a word.
Undergone.
Undergone. Undergone, right?
Yes, ma'am.
No, I don't know. Something doesn't feel right. Anyway, it went under, or anyway, meiosis happened to that. And because of that, 4 haploid microspore tetrad were made. in the case of the male. Now, in the female also, the megaspore mother cell will undergo meiosis and form the 4 haploid megaspores. But here the situation is a bit unique because out of the 4 megaspore, only 1 is functional. The other 3 are of no use and they degenerate, meaning they disintegrate. Okay. Then this functional megaspore would develop into the female gametophyte, that is the embryo sac. This method of embryo sac formation from a single megaspore is termed as monosporic development. So the kind of development in majority of the angiosperm will be monosporic development because it is happening from one functional megaspore. Mono means one, sporic is referring to the megaspore. So monosporic development. Clear? We'll keep it till here only. Now, you've understood this whole process of megasporogenesis. Is it clear? Do you have any doubts?
No, ma'am.
Okay. And the reason why the other 3 degenerate is not known. It's not known, but it is what happens. 1 is only functional out of the 4 and the other 3 would be non-functional and they would degenerate. Clear? Okay. Now you people can note down, and this is the last topic that we are discussing today.
Done, ma'am. Done.
Everyone's done?
Yes, ma'am.
1 minute. Done, ma'am.
All right. Okay, students, let's just keep it till here only for today. Do you people have any doubt? Anything that is troubling you?
No, ma'am.
Okay.
No, ma'am.
Okay, very
No,
good.
ma'am.
Okay. So, okay, now in the next class, we'll continue from how the embryo sac has been formed and how every cell, you know, sets at its place. And up until now, you revise your book. Revise the notes that you have taken down. And if you do have any problem, pinpoint it and then ask me in the next class. Okay. All
Yes,
right. Okay then. Okay then. I'll meet you students in the next class.
Thank you, ma'am.
Thank you, ma'am.
Good night.